So the first person to be diagnosed with Alzheimer's disease was Auguste Duta in 1906. And when asked a question, she'd often reply, "Oh God, I've lost myself. I've lost myself." So 100 years later, that remains one of the most poignant and eloquent descriptions of what it means to have this terrible disease. And the statistics for Alzheimer's disease, often called AD, are truly frightening. If you're expecting to celebrate your 85th birthday, you've got a nearly 50% chance that you'll be among the 14 million Americans that are expected to have the disease by 2050. And looking after you and your fellow AD sufferers is going to top $1 trillion. And it's not just the monetary cost. It's the terrible burden and the emotional price that has to be paid by the people that look after AD patients and their families. So I think an old Australian poster actually captures this very well. That is Auguste Duta. And this is that poster. It's an elderly lady sitting alone, and it says she has Alzheimer's disease. And then off to the side are a family, and it says, and they also suffer from it. I'm going to share with you some new and surprising findings that are coming out of our laboratory that may help us understand the origins of this terrible disease. But first, what is Alzheimer's disease? What did Alzheimer see when he first looked down his microscope in 1906? Well, the first thing he saw when he did unaided visual examination was a dramatically shrunken brain. But it was when he looked down his microscope that he saw the pathological telltales that would define the disease that was to bear his name. And the first thing he saw outside of cells were massive deposits of something that he named amyloid. And on closer inspection, these amyloid deposits resolved into structures with distinct morphological characteristics, and he called those amyloid plaques. Second, inside of cells, he saw thin thread-like structures called neurofibrillary tangles. So neurofibrillary tangles are found in a lot of neurological diseases, but huge accumulation of amyloid plaques is specific for Alzheimer's disease. And that's kind of how it stayed for the next 78 years. It wasn't clear what amyloid was made of. It wasn't clear why they accumulated in such extraordinary abundance in AD brain. But then in 1984 came a seminal paper that showed that amyloid plaques were made from a little protein called A-beta. And A-beta can self-organise into long, thin structures called amyloid fibrils. And it's these amyloid fibrils, a tangled mass, that actually makes an amyloid plaque. Visually, think a microscopic ball of fishing line. And those fibrils inside that tangled ball are all made of neatly stacked molecules of A-beta. So, in addition, we now know that the deposition of amyloid plaques triggers a series of pathological events, including neurofibrillary tangle formation, that ends with massive brain cell death. This is called the amyloid cascade model of Alzheimer's disease, and 85% of all drug development programs have tried to stop the amyloid cascade by targeting A-beta. There's been over 400 AD clinical trials testing some 250 compounds, but as yet, not a single one has emerged as able to slow the progression of this disease. Arguably, this is the worst performance in modern biomedical history. Some of the drugs failed simply because they were unable to modify A-beta, but a few did achieve their pharmacological objective, but still they failed to slow the disease's progression. We believe some of the reason for the failure of these therapeutic approaches is that there is a complete lack of understanding and knowledge about what the normal role of A-beta in brain is and why it forms fibrils. And the epiphany for us came when we encountered another little protein called LL37. And LL37 and A-beta share many important similarities. But LL37 isn't like A-beta, viewed as a pathological protein. The ability of A-beta to form fibrils is viewed as an accident of its chemistry. The A-beta protein itself is viewed as a piece of metabolic junk with an abnormal stickiness that causes it to form clumps that clump together into ever bigger masses and eventually result in amyloid fibril. And essentially, amyloid plaques are viewed as an accident of A-beta chemistry and the A-beta protein itself as an intrinsic bad player, and its misbehaviours are the root cause of Alzheimer's disease. But LL37 also forms fibrils, but it's not viewed as a bad player. And that's because LL37 is a member of a family of proteins called antimicrobial peptides, AMPs. And AMPs are natural antibiotics, and their function is to protect us against infection. They're the foot soldiers of innate immunity, the ancient arm of our immune system. And they're particularly important in brain because brain has evolved to exclude antibodies and other rough players in immunity that could damage their delicate structures. So one of the first things we did was an experiment to test whether A-beta has antimicrobial activity. And this is a classic antibiotic assay, and the absence of a microbial growth button confirms that A-beta does indeed have antimicrobial activity. Very potent ones. In fact, for some pathogens, A-beta turned out to be 100 times more potent than penicillin. But the really interesting thing, and the thing most relevant for Alzheimer's disease, is how it does it. Because microbes are captured and killed by amyloid fibrils. And what I'm going to show you next is the model that we've worked out in the last 7 years of how A-beta protects the brain from infection and the consequence of that, which is the deposition of amyloid plaques. So the first step in this process is the binding of clumps of amyloid of A-beta to the surface of microbial cell, in this case yeast cells. That binding triggers the fibrillization, which is the formation of an amyloid fibril. And the fibril shoots out from the surface of the microbe. That growing fibril and the clumps of A-beta on the surface of the microbe prevent the pathogen from attaching to our host cells. And if it can't attach to our cells, it can't infect us. And an unattached microbial cell is highly vulnerable, and those growing fibrils eventually tangle up around it and pull it into a compact structure called an agglutinate. And in that agglutinate, the pathogens are effectively neutralised, at least temporarily. You can actually see this happening down the microscope. And what I'm going to show you next is a figure from an experiment where we added pathogenic yeast cells, Candida albicans, to human host cells growing in cell culture. And normally the yeast cells go straight down, attach to the host cells, penetrate it and kill them. But if those host cells secrete A beta into the media, then that doesn't happen because they never get a chance. So this figure shows the candidas, which are the dark shadows, and you can see between them long, thin lines, which are actually the A beta fibrils. And that analogy I drew to a fishing line earlier on, actually not that far from the mark, because like fishing line hooking a fish, the amyloid fibrils hook the microbes and slowly they reel them in and they pull them down into this, an agglutinate. But A beta's job's not done there because microbes can escape from agglutinates. So at that point, in this compact, tight mass of fibrils and microbes, A beta's killing activities come to the fore to turn that agglutinate into a microbial killing ground. And the first thing it does is the fibrils form pores in the surface of the microbe, essentially holes that allow the contents of the cell to leak out. The bug bleeds to death. This is a classical AMP mechanism for killing microbes. But that's not A beta's only trick. So when a fibril forms from A beta, the protein's ability and affinity for copper ions goes up 1 billion-fold. So what that means is that the aggregate, the agglutinate, draws in copper ions from the brain milieu, and A beta uses that metal to catalyse chemical reactions that produces hypochlorite ion, better known as bleach. The bleach sterilises the aggregate, but it also does something else. At the high levels that are generated inside that agglutinate, it causes amyloid fibrils to fuse in a process called crosslinking. And that has two effects. The first one is to close down A beta's ability to generate more hydrogen peroxide, which is a good thing. If it was to keep going, it would damage the surrounding brain tissues. But the second thing it does is that it makes amyloid fibrils highly resistant to enzymic degradation. And that's important because the microbes secrete a whole bunch of different enzymes whose function is to degrade the fibrils and allow them to escape. So they're permanently entombed. Unfortunately, it also means that the brain's housekeeping cells have a very hard time removing A beta and the microbial agglutinate, which is why some of these plaques can be in your brain for 10, 20, 30 years. So as this next image shows, this process is very effective at killing the trapped microbes. This is a picture of Salmonella bacteria entrapped in an aggregate, an agglutinate, and you can see the cells look deflated and wrinkled. They're deflated just like a punctured football, and their surfaces are highly damaged from the bleach. So this works really well. This whole pathway protects the brain from infection. But the downside is these aggregates accrue in the brain and end up being amyloid plaques. And this is a picture from an Alzheimer's disease brain of a mouse that was infected with herpes simplex 1 virus 3 weeks earlier. And you can see that you don't have to be a neuropathologist to see that the virus and fibril structure generated looks a lot like this. The same thing Alzheimer saw when he peered down his microscope over 100 years ago. This model of how A betas fights infection is a rational framework on which the apparently diverse and often bizarre activities that have emerged from about A beta can be rationally understood. It also reveals why a lot of the drug trials didn't have much chance of working that targeted some of these activities. But for most non-scientists, the explanation of A beta's behaviours is a little esoteric. The most interesting and significant implication of our work for most people is this. If A beta is an antimicrobial peptide, plaques are generated in response to new pathogens, is Alzheimer's disease caused by a chronic brain infection? And our results do suggest that. But I would caution against concluding this is the smoking gun for infection causing Alzheimer's disease. There's a lot of inappropriate innate immune responses out there. And some of them involve the targeting of infections that aren't actually there. And AD may turn out to be one of them. Nonetheless, I have to say that the idea that infection can cause Alzheimer's disease is not as outlandish or as novel as you might expect. The first guy to suggest infection is linked to Alzheimer's disease, Alzheimer. And in fact, before the bad player A beta model, it was a widely held belief. But now, the current models, you don't need infection to explain Alzheimer's disease, because if you've got bad player A beta whose behaviours produce Alzheimer's disease, it explains it all. There is a small but dedicated group of scientists that have painstakingly assembled over decades an impressive and convincing body of data that suggests that infection is linked to Alzheimer's disease. And that data, along with their own findings, really is causing a sea change in the way this disease is viewed. And that has led to the consideration of anti-infectives as treatments for AD and an explosion of interest in innate immunity as potential pathways that can be targeted by drugs. So while the early promise of A beta targeting drugs seems to be fading, there's lots of reason to be optimistic. And there's an emerging consensus that a magic bullet, single magic bullet, is not going to work. You're probably going to need multiple drugs at different stages of the disease. What we're calling the 3 R's: right time, right place, and right treatment. But right now, progress on those new ideas is a lot slower than it could be, and a lot slower than it should be. And the reason is that AD research is cash constrained. For every dollar spent on Alzheimer's disease care, less than half of 1 penny is spent on AD research. And in 2018, while that funding level is actually 3 or 4 times what it has been for most of the last 20 years, it's still well below the level of other major diseases. Our lab has been very fortunate. We are one of the few labs with the resources to be able to pursue these new and novel ideas. But for most labs, that's not the case. We're funded by foundations like the Cure Alzheimer's Fund and Good Ventures that are organised around a venture capital model of philanthropy. And they seek out and invest in high-risk projects that also have the potential for high returns, not in money, but in knowledge. But for most AD researchers, the vast majority, they rely on federal grants. And the federal grants available at the moment are just insufficient to support the exploration of these ideas in the way that they need to be done. And it has to be said, if we had not got this alternative funding, I would not be here talking about these discoveries today. And it also has to be said that these discoveries They should have been made 15 years ago. What we've lost because of underfunding, we'll never know. But you've got to think, where would we be right now in terms of a cure if we'd got the funding we'd needed in the last 20 years? And where will we be if we don't get the funding in the next? It's very frustrating. And if we were to take 2018 funding and increase it tenfold and sustain that for 20 years, and that managed to advance our progress towards a cure just 1 year, just 12 months, it would pay for itself. And more than that, that year would save the lives of more Americans than were killed in the Second World War and every war since. So, funding for Alzheimer's disease is administered by the National Institute of Health, NIA. But the budget is set by Congress, which brings me to advocacy. So the role of advocacy in the development of effective treatments for HIV is a resounding example of collective determination and the power of persistent public pressure. But AD patients, AD sufferers can't advocate for themselves. As Auguste Duta said, I have lost myself. I have lost myself. It's up to us to advocate for them. And I'd add, it's not that we're just advocating for them, we're advocating for us as well. Because if you remember back to that 85th birthday statistic, if you and your partner are planning to celebrate your 85th birthday, there's a very good chance that you're going to do it bereft of a lifetime of shared memories. So I don't want to be all gloomy and doomy here at the end, because it isn't like that. The science has never looked brighter, but this disease has killed millions, and it will kill more and more and more millions if we can't pull together as a nation and confront what is a modern scourge that's stealing our loved ones. Thank you.